All right. Hey, everybody. Welcome to our monthly Brainstorm Live. I have a quick community announcement to make, and then I'm going to hand it over to Emily with an introduction of our featured author today. So the American Counseling Association does awards every year, right? And I received notification a few months ago that someone wanted to nominate me for one of those awards, but really not me. It was really an offshoot of the Brainstorm community. So I just wanted to say that I was the recipient of that award, but the reason I wanted to bring it up today was because there's been so many people throughout the history of Brainstorm that have had such an influence in its creation, to its administration, to all the lovely authors that we've had. So I wanted to share that announcement for you all and just say thank you, because it's your contributions that make that stuff happen. And even though my name is on the recipient, it is in no way, shape, or form my award. I do believe it's our community's award. So I wanted to share that with everybody. Just a quick announcement. This is the first time anybody's probably heard it. I haven't even told Emily yet. So thank you all. And Emily is one of a long history of coordinators. And Emily, your influence will come to an end soon as you graduate and move on from the program. So I also wanted to take this moment to say thank you, Emily, for all your coordination. And I hope the community will also share their appreciation as you transition on to the next phase of your career too. So with that, I'll hand it over to Emily.
Wow, thank you. That's wonderful news. Again, first I've heard it. Well, welcome back everyone to Brainstorm Live. Oh, thank you. I'm so pleased to introduce to you all Dr. Nafissa Ismail. She will be telling us about the impact of stress and hormones on the brain. So we're going to take the first hour to talk about for the presentation. And if you do have questions, please wait for the end. We will be doing the Q&A discussion where I know sometimes we do like interactive, but for this one, we're going to wait till the last half hour to talk about the questions. And I will also be sending the link for the CE certificates down in the chat close to when the hour is up. So keep a look out for that. And without further ado, I'm going to introduce you all to Dr. Ismail. Thank you.
Thank you so much, Emily, for the generous introduction, and congratulations to the entire team, to Eric, to Emily for the wonderful award. And thank you all for having me share with you the work that we've been doing in my lab. It's really, truly an honor to be able to speak with you about the impact of stress and hormones on the brain and what implications these have for mental health. So depression can affect anyone. It is one of the most widespread illnesses, often coexisting with other serious illnesses. Currently, depression is the third leading cause of disability worldwide. However, in January 2020, the World Health Organization published a report in which they predicted that depression would move to first place and become the leading cause of disability worldwide by 2030. But now we know that with the current pandemic, there's been a sudden increase in cases of depression. And so we have a hunch that we'll be reaching that first place much sooner than 2030. So it's really important that we make rapid progress in understanding the pathophysiology underlying depression and developing treatments that are effective, and also developing preventative measures. The reality is that the pharmacological treatments that we have currently don't work in all cases. So it's important that we take the time to understand depression much more deeply than we have in the past. What we know, though, from the current literature is that depression appears to be linked to a dysfunction in monoamine neurotransmission. This has been studied most widely. The current pharmacological treatments that we have actually concentrate on normalizing monoamine dysfunctions, But at the same time, given that these treatments are not necessarily working in all cases, it leads us to think that there is more to the story of depression than simply a dysfunction in monoamine neurotransmission. For instance, we know that stressful life experiences, and here I'm referring to stressful life experiences that are enduring, that not only impact our stress system, but that are also going to elicit an immune response. Those type of experiences like psychosocial stressors, intimidation, so on, abuse, trauma, have been shown to be linked to depression and anxiety later in life. We also know that most cases of depression and anxiety start during puberty and adolescence. In fact, the mean age of onset of depression is around 14 years of age. And then we also know that once we reach puberty, girls and women become 2 times more likely to develop depression than boys and men. And that this is particularly more likely to happen in women when they are undergoing sudden changes in hormones, such as after a pregnancy, during menopause, etc. So in my lab, we are really interested in trying to understand what are these stressful life experiences that can onset depression during puberty and adolescence? Why is puberty such a vulnerable period to stressors and is so vulnerable to developing disorders like depression? And what makes women more likely to develop depression than men? We know that hormones play a role, but what our research suggests is that hormones do not act alone and they interact with several systems. For instance, one of those systems is the immune system. And one of the things that sex hormones are going to do is that they are going to impact our immune and stress responses when we are faced with a challenge. And we do find these sex differences in immune response to pathogens. For instance, we do find that females are more likely to develop certain infectious diseases and autoimmune diseases, whereas males are more likely to suffer from other infectious diseases and non-reproductive cancers. But what's interesting is that this sex difference in stress and immune response is not maintained throughout the lifespan and changes actually as we age, such that it's true that after puberty, females tend to mount a stronger stress and immune response when they're faced with a challenge. But what we see is that prior to puberty, it's actually the males that will mount a stronger innate and adaptive immune response when they are faced with a challenge. So there's been quite a bit of research examining sex differences after puberty and prior to puberty, but there's been little research trying to understand what the sex difference is in immune and stress response during puberty. And so this is why we're so interested in focusing on that period, because we know that puberty is a critical period of development. It's during this period that we are going to find that our hypothalamic pituitary gonadal axis is going to become activated. This will lead to the maturation of the reproductive system and the production of gonadal sex steroid hormones. These sex steroid hormones are then going to contribute to the remodeling and reorganizing of the brain during puberty and adolescence, and it will do so in a sex-dependent manner. It's also going to then lead to changes in behavior that are also going to be sex-specific, and also sex-specific changes in physiology. But when we are exposed to a stressor during puberty, we find that there is an inhibition or downregulation of our HPG axis. And this then leads to a suppression of the production of the gonadal steroid hormones during such a critical period of development. So we can understand that during a period where these hormones are so important, when stress is on board and we shut down the production of these hormones, it can impact the maturation of several systems, including the sex-specific development of the brain. And so we're wondering if that could then link perhaps to the development of mental illnesses early in life, such as during puberty and adolescence. And the model that we are thinking is this one, where there is this bidirectional communication between hormones, stress, and inflammation. And those systems then bidirectionally impact mental health, such as they can contribute to the development of mental illnesses. And once mental illnesses are developed, there is going to be communication back to the hormonal system, leading to further changes in hormone production, also impacting future stress reactivity and inflammation, essentially creating this vicious bidirectional circle. So we wanted to examine really the fundamental mechanisms that underlie this bidirectional vicious circle. And so we wanted to use mouse models that would allow us to get to these fundamental mechanisms. And so in my lab, to induce stress and inflammation, we use lipopolysaccharide, which I'm now going to refer to as LPS. And LPS is a bacterial endotoxin that is isolated from the cell wall of a gram-negative bacteria. And in our case, it's the E. coli bacteria. And when it is administered systemically, LPS will bind to toll-like receptor 4 that is located on the surface of immune cells. And once it binds to this receptor, it will activate an intracellular cascade of events that will then lead to the production and release of pro and anti-inflammatory cytokines. And these cytokines that are going to be produced are not just going to remain in the periphery, in the body, causing inflammation, but it can also then get in the brain and cause neuroinflammation. And it does so either by just creating the production and release of cytokines within the brain, or by compromising the integrity of the blood-brain barrier and allowing infiltration of these inflammatory markers from the periphery into the brain. So with my research team, we were interested in examining whether we can create a mouse model of pubertal stress that would allow us to better understand how stress during this critical period of development can induce depression, specifically in females. So in this study, we had mice arrive to our lab at 3 weeks of age, just as they were being weaned. This period is known to be a stress-resistant period. And then we let the animals age and acclimate. And once they reach 6 weeks of age, which is a stress-sensitive period during puberty, we expose them to either saline or LPS. This made the animals sick for about 48 hours, but then they recovered and they look completely fine. We let them then reach 10 weeks of age, adulthood, and once they had reached adulthood, we then exposed them to various behavior tests. So before I show you the results that we obtained on the depression-like behavior, let me first just explain what we found acutely at 6 weeks of age when we administered the LPS treatment. We found that when we administer LPS, we noted significantly more sickness behavior, changes in body temperature, an increase in pro and anti-inflammatory cytokines, and more activated microglial cells in males compared to females. But females showed an increase in serum corticosterone compared to males. Now, we also found important age differences when we administer LPS at 6 weeks of age versus at 10 weeks of age in adulthood. For instance, we find that when we give the LPS in adulthood, we note greater sickness behavior, greater changes in body temperature, more serum pro-inflammatory cytokines, and more serum corticosterone compared to mice treated with LPS at 6 weeks of age during puberty. So it seems to us that puberty appears to be a period that is hypo-responsive to stressors and immune challenges. And that makes sense when we think about it, because during this period, the stress system and the immune system are still maturing and developing. And so when they are faced with a challenge, it's sort of like they are unable to process it and deal with it. And that's really unfortunate, because it seems to be maladaptive, because it's these pubertal animals then that are showing enduring changes in behavior, whereas the adults are not. So we see that this way of coping with the stressor during puberty is actually maladaptive. For instance, we find that animals that are treated with LPS during puberty display reduced sexual receptivity, reduced performance on spatial learning and memory tasks, reduced performance on dopamine sensitive behaviors, greater anxiety-like behavior, and greater depression-like behavior. So today I'm going to focus on talking about the data that we got on the depression-like behavior. So to examine depression in our mice, we use the 4-swim test. It's a test that is considered to be valid in the literature, although there are some debates. But it's the only test that we have at the moment that is really measuring behavioral despair in the mice, the way that would resemble what we're seeing in humans. So the way that we do this test is that we have these beakers of water that we fill with room temperature water, and then we let the mice swim for 5 minutes. And what we see is that the mice are going to swim and then transition from series of swimming to trying to climb to then immobility. And when they're immobile, they assume a posture like the one you're seeing here. And this immobility is what we quantify, this duration of immobility, and it is indicative of the behavioral despair symptom of depression, such that the greater duration of immobility we would find in animals, the more behavioral despair and depression-like behavior we would be observing in the mice. And what we noted is that when mice are treated with LPS at 6 weeks of age, they display longer durations of immobility compared to their saline controls, suggesting that the LPS treatment is increasing depression-like behavior in females that are treated with LPS at 6 weeks of age. So we were really excited by these findings because it allowed us to develop this animal model of increased susceptibility to depression in females. We also noted that this effect was really limited to the females because we didn't find increased depression-like behavior when we administered LPS to the males. And we also didn't find this increased duration of immobility when we treated adults with the LPS. So it was really limited to the pubertal animals. We then wanted to examine how all this interacts with hormones and whether hormones can mitigate or increase the effect of LPS on depression-like behavior. One thing we know is that when we overreactamize mice, we do tend to see an increase in immobility in depression-like behavior. But when we treat these overreactamized mice with estradiol, we then mitigate the duration of immobility. So here, we overreactamize the mice in adulthood. And what we see is that the saline-treated animals displayed long duration of immobility as expected. But when they were given then estradiol, we saw a reduction in immobility. But in the mice that were treated with LPS at 6 weeks of age, when we gave them the estradiol, we see that the estradiol was unable to reduce the duration of immobility. So it was unable to decrease depression-like behavior. But that was limited to 6 weeks of age, because at 10 weeks of age, we see that estradiol treatment is really effective at reducing depression-like behavior. So we're really intrigued by what exactly is changing when we give LPS or stress during puberty. At the moment, this model summarizes what we're seeing. So it has been shown previously in the literature that when we overreactomize mice, we then increase the duration of immobility, so increase depression-like behavior. But when we take overreactomized mice and treat them with estradiol, previous literature has shown that there is then a decrease in the duration of immobility. But what our work shows is that when we treat these overreactomized mice with LPS at 6 weeks of age, estradiol treatment is no longer effective at reducing the duration of immobility. So we were interested in understanding why that is the case and what is happening here and why LPS would prevent estradiol from being able to alleviate symptoms of depression. And so to understand that mechanism, we moved our attention to the gut-brain axis, because we know that there is a bidirectional communication between the gut and the brain, and that the microorganisms in our gut can influence stress and immune responses. So we wanted to see if exposing the gut to good bacteria, such as probiotics, could perhaps mitigate the effects of stress on depression-like behavior and perhaps reactivity to hormones later in life. So to do this work, we partnered with Leaky, which is a small family-owned company based in Quebec, Canada. And we were funded by the Canadian government to do this work by an Insert Engage grant. And essentially, this company produces and commercializes probiotic products under the brand name Gourmet. And so it is products that are readily available in health stores. And this is one of the reasons why we chose to go with this company, because we wanted to use a probiotic that is being used by humans, that has good ecological validity, that is easy to prepare, and also that is well quantified in terms of the different types of bacteria, probiotic bacteria that we find in there, which we found that, you know, often you're going to find on the market probiotic products that are not well detailed in terms of their content. So Leaky and their Gourmet products were really good that way. And so we partnered with them. We used their kefir. And so in our study, we again had mice shipped to us from Charles River at 3 weeks of age as they were being weaned. And then we let them acclimate, and at 6 weeks of age, they were treated with saline or LPS. But starting 1 week before the saline or LPS injection and continuing 1 week after, so for a total of 2 weeks, the mice were given either a controlled broth or the kefir probiotic mixture. And then we let them age and we examined their behavior once they reached adulthood at 10 weeks of age. And what we found is that actually the pubertal treatment of kefir prevented LPS-induced depression-like behavior in females. So that was really, really exciting for us, because it felt that we are on a path going towards finding a potential preventative measure for stress-induced depression onset during puberty and adolescence. But of course, there is a lot more work here left to do. So we wondered what exactly is happening at the level of the gut when we administer our LPS treatment and when we give the probiotic treatment. So we noted a number of changes, age-specific, sex-specific changes. So what you're seeing on the left is a heat map depicting age differences in LPS-induced changes in bacterial genera 24 hours after the LPS treatment in both males and females. And on the right, you're seeing a table depicting sex differences in LPS-induced increases or decreases in about 27 genera. But if we go up the taxonomic hierarchy and examine things at the level of phyla, what we find is that in males, when we administer LPS, we find a significant decrease in Firmicutes and a significant increase in proteobacteria. Firmicutes is typically one of the most abundant phyla in the mouse and human gut, and it is involved in energy reabsorption. So given that our mice are experiencing weight loss, they lose about 10% of their body weight after the LPS treatment, it makes sense to see that there is a decrease in Firmicutes. Proteobacteria is typically found in low concentration in the body, but what we find is that it tends to increase when there is inflammation in the body. And given that with LPS, we do find an increase in inflammatory markers, it makes sense to see the increase in proteobacteria. However, when we administer kefir 2 weeks around the time that we give the LPS, we find that there is no longer an increase in proteobacteria, but instead we find an increase in verichromicrobia. And verichromicrobia is a phylum that we generally find in low concentration in the mouse and human gut flora, but we do see it increase when humans are under antibiotic treatment. So it's possible that it is related to enhanced immune function, and that's maybe what the probiotic treatment is doing, is enhancing the immune system. But we do find that all these changes that we're seeing within the gut are only transient. So we find them up to about 1 week following the immune challenge and the kefir probiotic. But after that, the gut seems to recover and everything seems to be back to normal. But it's possible, though, nevertheless, that these short-lasting changes, even just a week, is sufficient to cause enduring changes then in the brain and perhaps then predispose to mental illnesses. In the females, we find slight differences. So we find that when we administer LPS, we see a decrease in Firmicutes again, just like with the males, but we find an increase in Bacteroidetes. Bacteroidetes is one of the most abundant phyla in the mouse and human gut, and it is linked to undernutrition and weight loss. So when there is weight loss and undernutrition, we do see an increase in Bacteroidetes. And given that our mice did lose about 10% of their body weight in the first 48 hours, it makes sense to see that increase in Bacteroidetes. However, when these females are treated with kefir, we no longer see the decrease in Firmicutes or the increase in Bacteroidetes. Again, these effects are not long-lasting. They last for about a week, but it just tells us how even a week of change in the gut microbiota could have effects on brain function. So what are the possible mechanisms underlying all this? We truly believe that it's linked to a leaky gut leading to a leaky blood-brain barrier. So that when there is changes in the gut microbiota and there is gut dysbiosis, there is thinning of the gut lining that becomes more permeable and leaves out toxin, allows toxins to leak out of the gut. And these toxins, once they leak out of the gut, they then eventually reach the brain through the vagus nerve and compromise the integrity of the blood-brain barrier, then allowing infiltration of these toxins within the brain and causing neuroinflammation. So you might have heard that what happens in Vegas stays in Vegas, but I can tell you that this is incorrect. What happens in the Vegas actually gets to the brain and compromises our blood-brain barrier and causes neuroinflammation within the brain. And we've tested that. We've tested the impact that LPS has on the integrity of the blood-brain barrier. And what we find is that 24 hours after the LPS treatment, we do find increased permeability in the blood-brain barrier of females treated with LPS. We don't see it in the males at 24 hours, but we do see it at 1 week. We do see that at 1 week, the males are showing increased permeability. The females seem to have recovered. So there is a little bit of a lag in when we're seeing that change in blood-brain barrier permeability in the males compared to the females, but it does seem to be happening in both, just at slightly shifted time for, again, a limited period of time. So it's a transient effect. After that, we do find that the blood-brain barrier recovers from the stressor and is no longer more permeable. But you know, again, these short transient changes during critical periods of development may be sufficient then to increase the vulnerability to certain mental illnesses like depression. So in terms of the mechanism, so going back to this bidirectional vicious cycle that I presented to you earlier, what we're now thinking is that in addition to stress and hormones, we also have the gut microbiome impacting, again, in a bidirectional manner, stress and inflammation, hormones, and then impacting the brain and the vulnerability to mental illnesses. So what are some of the implications of all this? So we're still, of course, working on all this. There is a lot of research left and a lot of work for us to do in this area. But one of the applications that we found is with oral contraceptives. So we do know that oral contraceptives are one of the most widely used methods of reversible contraception. They're made of synthetic estradiol and progesterone, and we've been using them for many, many years, at least 60 years. And currently around the world, there is more than 100 million women using oral contraceptives. A lot of women complain of experiencing mood changes, irritability, depression, increase in premenstrual syndromes. And we see that in about 10% of the women taking oral contraceptives. And about 30% of these women are then just simply going to discontinue using oral contraceptives because of the side effects on their mental health within the first 3 months of taking them. But when we look in the literature, we really find a lack of research and a lack of consensus on what exactly oral contraceptives are doing to the brain and whether there is any link with mental illness. And this lack of research and consensus comes from a bigger problem, actually, where we do see in the literature a significant lack of research on issues related simply to women's health in general. So we really need to push research related to women's health issues as much as possible. We are seeing some progress now, but there is still a lot of work left to be done. So one of the questions that we asked ourselves is whether oral contraceptive usage during puberty could increase the vulnerability to mental illnesses. Because we do know that a lot of girls start taking oral contraceptives very early in development, sometimes during puberty, just as they're starting their menstruation. And it's not necessarily for contraceptive purposes, but rather to regulate hormone-related problems such as acne, premenstrual syndrome, heavy menstruation, etc. And given that this period is also linked with increased mood disorders, we're wondering whether there might be a connection between the two, because we do know that once we start taking oral contraceptives, these synthetic hormones are going to downregulate our hypothalamic pituitary gonadal axis, such that we will no longer be able to produce the endogenous hormones that we typically do. And knowing that the endogenous hormones are responsible for so many changes in the brain, in the physiology, during puberty and adolescence, leads us to think that perhaps we're leaving now this development and maturation to those synthetic hormones that are found in oral contraceptives. So we first asked whether oral contraceptive usage during puberty affects brain structure. And in this study, we had participants that were taking oral contraceptives and that either began taking them during puberty, so within 6 months of starting their menstruation, or in adulthood. We also included naturally cycling women that we compared at various stages of their menstrual cycle to try to catch them at varying concentrations of hormones, estradiol and progesterone. So we had women that we tested during the early follicular stage where they typically have low hormone concentration. Then we also tested women during the pre-ovulatory phase where we would expect high levels of estradiol but low levels of progesterone. And then in the late luteal phase where we would typically see high levels of estradiol and progesterone. And so when we examine brain structure in these females, what we noted is changes in gray matter volume. So we found that women that were on oral contraceptives displayed reduced gray matter volume in the right putamen and the left post-central gyrus compared to naturally cycling women. And what we see is that these areas, like the right putamen, is a component of the basal ganglia, an area that is involved in movement, in reward, and so on. And so it's interesting to see how the decrease in gray matter volume might impact the function of the putamen in these women. The post-central gyrus houses the secondary somatosensory cortex. And so it appears to play a role in the integration of somatosensory stimuli and also in memory formation. So we don't yet know how the decrease in gray matter volume could impact those functions of the left post-central gyrus. So that remains to be investigated. We also noted changes in white matter volume. So we found that women that were on oral contraceptives displayed an increase in white matter volume in the right putamen, where we had previously seen a decrease in gray matter volume. It's almost like because there is a decrease in gray matter volume, the brain is trying to compensate with an increase in white matter volume. But we also found an increase in white matter volume in the parahippocampal gyrus compared to naturally cycling women. And we also found that women that began taking oral contraceptives during puberty showed an increase in white matter volume in the right fusiform gyrus. And the right fusiform gyrus is part of the temporal and occipital lobes, and it appears to be involved in language processing. And so it's unclear again how the increase in white matter volume might be impacting the function of the right fusiform gyrus. We did explore a little bit the impact of oral contraceptives on brain function, but there is still a lot of work to be done in this area. But in our study, we used a unique NBAC task that actually we developed just in our lab. It was an NBAC task that contained neutral, positive, and negative pictures. And participants had to remember and recall whether they had seen this picture 2 sets ago or 1 set ago. And we did find differences in performance based on the nature of the images, whether they were simply neutral images like the cupboards that you're seeing here, or if they were negatively charged images such as abused animals, car accidents, or whether they were positively charged images, depicting happiness and so on. And what we found is that women that were on oral contraceptives, they displayed greater brain activity in the right inferior frontal gyrus and in the left lingual gyrus when they were exposed to images that were negatively charged compared to naturally cycling women, sort of making it seem that women that are on oral contraceptives tend to react more to negative stimuli. Now, going specifically to women that began taking oral contraceptives during puberty, we found that in these women, there was increased activity in the right insula and the right precentral gyrus when they were asked to recall images that were 2 sets back versus images that were simply 1 set back, making it seem that processing images that were 2 sets back required more effort, perhaps from these brain regions, and this could be why we are seeing increased brain activation. But again, of course, all this needs to be further investigated. We also wondered whether there was any change in resting state functional connectivity in women that are taking oral contraceptives versus those that are naturally cycling. Resting state functional connectivity refers to the fact that our brain is always emitting signals, and even when we are at rest and we're not doing anything. But recent literature has shown that this activity, this brain activity that we see at rest, is different in depressed patients versus those that are not diagnosed with depression. And so what we found in our study is that women that are on oral contraceptives, they show increased resting state functional connectivity in the salience, central executive, and reward networks compared to naturally cycling women. And they also show increased resting state functional connectivity between the putamen and the frontal cortex. And if you recall, the putamen is the area where we found decreased gray matter volume and increased white matter volume. We also found that specifically in women that began taking oral contraceptives during puberty, there was an increase in resting state functional connectivity in the salience network compared to those that only began taking oral contraceptives in adulthood. So this was really interesting because the increased resting state functional connectivity in the salience network has been previously linked to depression. And we do find that patients that are suffering from depression show an increased resting state functional connectivity, specifically in the salience network, compared to those that don't express any symptoms of depression. So it's still unclear whether this increasing resting state functional connectivity in women that began taking oral contraceptives during puberty is in any ways linked to depression. But here in our study, none of the participants had received a diagnosis of depression. So these are normal women. It's not a clinical population. So to conclude my presentation, what our work has shown so far is that there are important age and sex differences in acute response to an immune challenge. We also find that exposure to the immune challenge causes enduring sex-specific changes in behavior. For instance, in the females, we have seen increased depression-like behavior. In the males, we have seen increased anxiety-like behavior, but I didn't show you that data today. We've also found that LPS treatment seems to transiently alter the gut microbial composition in a sex-specific manner. And it also seems to cause transient increases in blood-brain barrier permeability in males and females, although a bit shifted in time. We do also find certain changes in receptors within the brain, particularly in males. But what we do find is that treatment with probiotics does seem to protect against the enduring effects of stress, particularly on depression. And in terms of implications of all this, well, we do find that the presence of synthetic hormones that we can find in oral contraceptives appear to cause changes in brain structure and function, and that there are some of these changes that are particular to taking oral contraceptives early in development during puberty. So, of course, there is still a lot of work for us to do to understand all these systems and how they interact with one another to increase our vulnerability to mental illness. But we are really hopeful that one day our work will help us better understand the risk factors for mental disorders during puberty and hopefully provide potential preventative measures. So with that, I'd like to thank all the graduate students in my lab, the undergraduate students, and the international visiting students, all the collaborators, our funding agency, the MICE for their contribution, and you for your time. Thank you.
Thank you for thanking the mice. I love that little I love that little throw in there at the end.
Yeah, we do appreciate, we do see animal research totally as a privilege. And I think that it's important to recognize that.
There's a statue, I think it's in St. Petersburg in Russia, of a mouse, and it's to dedicate the lives of all the mice that science. It's pretty cool. It's a cool fact. But thank you so much for your wonderful presentation. I'm
My pleasure.
If you could say more about sort of the role of probiotics, and like, is it just during puberty where they have that effect of protecting against your vulnerability, or is it like throughout the lifespan, or is it just puberty?
Yeah, no, that's an excellent question. So we've only given the probiotic to our mice during puberty, and specifically during the 2 weeks around the presentation of our stressor. We haven't tried whether giving it in adulthood would still result in an alleviation of those symptoms of depression. But we were nevertheless really happy to see that simply a 2-week course of probiotics seems to have these enduring effects in preventing depression-like behavior. But absolutely, one of the next steps we wish to do is examine the impact of probiotics later in life.
Thank you, Emily, for asking that question. I have had a similar question, and that is this. I work a lot with people on the autism spectrum, and there is, okay, Nafissa, you know where I'm going with this.
Yes.
But my question is there anything that increasing the microbiome, the way you're describing, that it doesn't support, it doesn't help? It seems like it's a no-brainer. There's so many areas where when the gut is, out of balance, it affects so many different things, and it's tied to so many different adverse conditions. So
Absolutely.
should we all just take that stuff?
Yeah, no, that's an excellent question. I mean, probiotics have very few side effects. I mean, from what we know, other than constipation, and that too is mild constipation that lasts for about the first month to 3 months of taking the probiotics. After that, individuals no longer seem to be experiencing constipation from taking probiotics. Why not take them on a regular basis anyways? But the point is that in some cultures, we do see that probiotics have been integrated quite a bit in terms of treatment prevention, but also in treating certain disorders. So for instance, I have colleagues in China and Japan that are using probiotics to treat cognitive decline in aged adults that are displaying decreases in cognitive functioning, and they are noticing improvements. But here in North America, probiotics are not naturally part of our diet, and so it's a little bit more difficult to integrate them. And I think that for us, like, we need a lot more research and findings that support the use of probiotics. So there is work on the way, and as you've said so nicely, there's been great improvements of probiotics on symptoms of autism, and we found it also on other disorders as well. And so the work is coming, and then hopefully eventually we'll be at the level where we can say, that probiotics are really helping with so many diseases.
I have a question. Sorry, Dr. Beeson, you go ahead.
No.
Okay, so I have a question, and it's I'm not sure I'm going to articulate it properly, but is the research coming about, because this looked at discrete genders, women and men, and we know depression is so high with transgender people. I'm not even crossing the others, extending the other gender issue, concern, differences, definitions. But is the research coming about this kind of the biology and the physiology with transgender persons and depression?
That's an excellent question. We definitely need more research in that field. You are so right. For us, I would say we're just starting now to understand the interaction with hormones. And so in my lab, we focused on males and females, and even examining these sex differences is still at an early stage, so we definitely need more research in this area, and also in transgendered individuals. I know that there are labs that are studying it. We haven't been doing so yet at the fundamental level, but we would love to understand more about the mechanism so that we can then start integrating those aspects as well in our research.
Thanks for that, Keishan, because I think that extends a little bit further, because you're focusing primarily on sex differences. So I think that defining those, like defining the variable, is really important in research. And then I guess this is just my own curiosity. Are there animal models for transgender differences?
So not equivalent to the way that we construct it in humans, right? Like but we do find that, for example, animals will display behaviors that are more homosexual versus heterosexual, and we've seen that too in mice and rats. And so there is definitely that range that we need to explore more. It's not as common, like when we get a batch of mouse, we're not necessarily gonna see it in great proportion, but we do need to explore that more, so I do think that it is present in various animal models, but it's understudied. Yeah.
And, I know that some animal models are bred to display certain traits or whatever, so it's interesting to think about sexuality and gender in an animal model, which, my curiosity is just now being piqued. So Keishan, thank you for asking that question, because now I'm like, we know that we don't do it enough with human models, but now I'm thinking about what's the animal correlate of those studies. So this is a bit off of that. So does anybody else want to jump on the gender sex conversation before I pivot? We all want to. It's just, where do we articulate that, and how do we really look forward? But I was thinking, you mentioned about isolating the kind of strain of probiotics, and that was the reason you picked the one, I guess, producer of the, I forget what you called it. Pardon my.
Leosan, the company. Yes. Yeah.
And when I do my own research on probiotics personally, like, it's really hard to isolate, what specific effect am I looking for? But some of that research is there. This strain is best for this. This strain is best for that. So I guess, thinking about your lab and maybe the future of the field, do you see it getting very, very discrete of focusing on a specific strain, or do you see it more an amalgamation of like 18.9 billion cultures in a commercial probiotic?
Yeah, no, that's an excellent question. We are definitely going to narrow down the specific probiotic strains, and we've begun doing that. So we started with the kefir, which is a cocktail of probiotic bacteria, but at least we had a product where they were all quantified, which is why we chose that product. But now we're moving to narrowing down specific strains. And so we're working with a different company that also sells their products like available for human consumption. And their products, so that company is called L'Allemande, and their products are made of 2 probiotic strains. So we've narrowed down to the lactobacillus type of bacteria, but even there are so many different types So the products that they sell have two types of lactobacillus bacteria. And so we're comparing two of their products. That research is currently ongoing, and we're hoping that we will be able to identify the strain. But so far, what our data is showing is that it's not one strain fits all, but rather that there are some strains that seem to work better with females and other strains that tend to work better with males. And now we're also wondering whether there is an age difference, and that maybe there are some strains that work better on the developing gut versus some others that may work better on the adult gut. And so it may not be that it's one probiotic fits all, you know? Yeah.
I may have missed it, but does your research follow or track like epigenetics